Yeah, actually,
Okay.
I was just talking to that to Esperanza, and yeah, it
Yeah.
was 2008. Yep.
Okay. I actually visited 2 times.
Twice, right? Yeah.
Yeah.
When was the other time? You don't remember?
Yeah. We'll have to do it again.
Yeah, by all means. So
That was very
what
interesting talking with Esperanza. Yep.
okay, good to know that. So what is the weather like there?
Oh, it's cold and snow. We have lots of snow. It's like living on top of a mountain in Mexico.
I can imagine. Well, a very high one, to be honest with you.
Yeah, very high one. Exactly. Yeah. How about in Cuernavaca?
Well, no, we are doing just fine. I mean, weather-wise, yeah, it's great. I have to
Always
say.
springtime, right?
Yeah, exactly.
Yeah.
All right. Do you want to start now, or shall we wait another minute or two?
I'm flexible. Whatever you think is good.
Because you need to leave at 1 p.m. sharp, right?
Yeah, gosh. Well, I don't know. Don't worry about it. I'll figure it out. I forgot about, what is that? Oh yeah, it's some graduate thing. Yeah, don't worry. It's fine. Yeah, we can start, I guess.
Okay. Right. I'm going to start. Well, hello, everyone. Thanks for watching Scott's seminar. It's my great pleasure to introduce Scott Edwards. Scott is professor of organismic and evolutionary biology at Harvard. He got his BA from Harvard University and his PhD at University of California, Berkeley. Scott was also an Alfred Sloan postdoctoral fellow in molecular evolution at University of Florida, Gainesville. He joined Harvard in 2003 as full professor, and his research focus on diverse aspects of avian biology, including evolutionary biology, biogeography, and comparative genomics. He was one of the first people that conducted analysis based on DNA sequencing regarding phylogeography. Scott has received many awards. Among them, he was selected Fellow of the American Association for Advancement of Science, and he's member of the U.S. National Academy of Sciences. Scott has published more than 160 papers and several of them in Nature and Science, and he has over 27,000 citations. Scott is also an associate editor for important journals like PNAS, or Molecular Phylogenetic and Evolution, and was also associate editor for Molecular Biology and Evolution and Systematic Biology. Well, it's my great pleasure to give you the one and only Scott Edwards. Scott, take it away.
Thank you very much, Santiago. And it's really nice to connect with Santiago and the CCG again. I visited a few times, maybe back in 2008. Anyway, I'm grateful for this opportunity to tell you a little bit about my research. I will share my slides here. Let's see here. Great, thank you very much. And there we go. Great, there's my talk title. And I am very much a natural historian. I don't do a lot of experiments in my actual research. And so I take advantage of natural experiments to try to understand connections between genotype and phenotype. And so I'll tell you about two examples of that. And the first one will involve a project that I'm actually working on with Santiago now. It involves a Mycoplasma bacterium that has infected house finches. And this is a really interesting system because we understand the recent evolutionary history of both the Mycoplasma and the house finches. Both of them were introduced to different parts of the United States. And so that I'll tell you a little bit about that. And then I will tell you about a story in deeper time on the bird tree, where we're looking at convergent evolution. And in particular, we will look at the loss of flight in birds. I'm an ornithologist, and so all of my projects involve birds. And this one I think you'll find interesting because it shows how you can use comparative genomics to try to find genes that are important for a phenotypic trait. Okay, well, let's start with the house finches. House finches are a widespread species in the Western US and in Mexico. And I will say right now that I really would like to come to Mexico and sample some house finches, because all of the studies that I'm familiar with really they neglect the Mexican populations, and we need to make sure to sample those to understand the true diversity. But you can see that it's been introduced to the Hawaiian Islands and to the Eastern US at different times in their history. And about 1994 or so, field biologists started seeing house finches that were sick, especially in the area of Washington, D.C., in the mid-Atlantic states. And they quickly figured out this was a Mycoplasma. And this map is meant to show how quickly the Mycoplasma spread across the eastern United States. And so my lab has been interested in understanding the evolutionary consequences of the Mycoplasma on the birds, as well as how the Mycoplasma itself has evolved. And now it's crossed the Rocky Mountains into the western part of the country. And so it's very widespread now. And we don't really know the status of this in Mexico. That's why we need to sample some birds in Mexico. Now, I would just summarize many of our studies over the years. We've taken advantage of different kinds of collections. For example, we have a very nice series of house finches collected before the epizootic in the late 1980s. And this was a really nice resource so we could compare the birds and their genetics later in time as we sampled in different parts of the country. You can see in the red here is where the Mycoplasma has been spreading. And now it's probably throughout the US, although we collected several birds in New Mexico just a few years ago, and none of them seem to have any Mycoplasma. And just I'll tell you a little bit about some of our studies then. And one thing we found was comparing birds in Alabama, where the pathogen has been for many years now, over 20 years, and then comparing those with birds in Arizona, where at the time of this study, there hadn't really been much exposure at all. And you can see that the Mycoplasma load is higher in the Arizona birds. It seems like birds in Alabama have evolved perhaps a bit more resistance. Again, just to remind you, Alabama is here in the east, whereas Arizona is here in the west. And we also found some really strong differences in gene expression of the host. So you can see here on the y-axis, we have Mycoplasma load, of individual birds. And then on the x-axis, we have basically what did its transcriptome from the spleen do when it was infected experimentally. And this showed that a lot of the birds that had low mycoplasma load ended up actually, their gene expression tended to increase more than did birds that had a very high mycoplasma load. Again, this is mostly contrasting Alabama birds, which are more on the right, and then Arizona birds, which are more on the left. So some very interesting short-term dynamics going on. And I thought I would just mention the mycoplasma genome work we've been doing, because this is what I'm now following up with Santiago. I'm very excited about this. But the Mycoplasma Galacepticum genome is it's larger than the genomes that Esperanza was just telling me about. Esperanza was telling me about her mycoplasma from the scorpions. Their genome is about 600 kilobases. Well, this mycoplasma is a bit larger. It's about 1 million base pairs. And you can see it's got a lot of variations when we compare the chicken strains to the house finch strains. And I'm looking forward to characterizing this better with some new PacBio data that we have, and working on this with Santiago and his postdoc, Gamma. And just some of the things we found were that the genetic diversity of the house finch mycoplasma strain was much, much lower than what we found in diversity among the poultry strains. So this mycoplasma ultimately came from chickens and turkeys. And you can see that their genetic diversity was much, much greater than the strains that we found from house finches, sampled over different time periods. And then I'll just also show you briefly a really interesting finding we had with the CRISPR loci of these mycoplasma. Whereas in the chicken and turkey strains, which you see here at the bottom, those strains had very large CRISPR arrays, many repeated units, and lots of variation, lots of turnover between different strains. By contrast, the house finch strains, by the time we got to 2007 strains, not only were the CRISPR loci decreased in number, you can see there are much fewer CRISPR loci, but the Cas genes, which are these black ovals here, the Cas genes had been completely lost. And so we're very curious why the CRISPR locus was degraded in these more recent strains. And we're interested in finding out the status of the CRISPR in the more recent strains that we'll be working on with Santiago. I'll tell you a little bit now about the birds. My graduate student, Allison Schultz, has been conducting low coverage sequencing of the birds at different time periods, both in the East and in the Western US. And we've tried to sample birds before an epizootic and after an epizootic, putting together our own field collections as well as museum collections. And we think we have some interesting signals, although it's still ongoing work. And so, for example, we can, with this genome data, we can clearly distinguish the birds from the Western US, which are here in green, and the Eastern US, which are on the left side in different colors. So that introduction to the Eastern US caused a strong genetic differentiation. We also know that there was some genetic diversity that was lost when the birds were introduced to the Eastern US. So for example, here is the original level of genetic diversity in the Western birds and in the Eastern birds before the epizootic. And you can see that not a lot, but a fair bit of reduction of genetic diversity in the Eastern US. Now, if we sample 10 generations after the Mycoplasma hit these two regions, you can see that genetic diversity declined in both regions very slightly, but detectably. And then finally, if we go 20 generations after the epizootic in the eastern US, you can see that the genetic diversity actually rebounded a little bit. So maybe some of these changes in genetic diversity are due to the spread of the pathogen, because we know, for example, that in some locations in the east, the house finches declined by up to 50%. So, you know, it was a detectable reduction in population size. We can calculate how much allele frequency change occurred between different places and between different time points. And so you can see here on the top, we're comparing 1990 to 2001, 1990 to 2014, and then 2001 to 2014. In general, the level of differentiation was very low. I think you're probably all familiar with FST. It's just it varies from 0 to 1, where 1 is very strong differentiation. And you can see that most of these values are very close to 0. So it's there's not a lot of genetic differentiation going on at these different time points. But we can try to begin to tease out regions of the genome that are showing unusual shifts in allele frequency. So for example, on the y-axis, we have a measure of basically the magnitude of FST between time points. And then on the x-axis, we have a measure of linkage disequilibrium, which measures sort of how correlated different places in the genome are with each other. And this basically in these plots, those points that are high up on the plot and a little bit to the right would indicate regions that are undergoing unusually large frequency changes and perhaps increases in linkage disequilibrium. And what you can see is there's a lot of genes in there. Clearly, these birds are shifting many different genes. But we actually see some genes that are of interest in terms of their immune response. So BBX up here in the middle panel is well known as a transcription factor that influences the T cell response. And so that's something we want to follow up on. And ENPP1, which you see on the left panel, that's interesting also from the point of view of the immune system. So these are things we want to follow up on. You know, we call what happened in the house finches probably is what we call a soft sweep, meaning the advantageous variants for resistance were already in the population. It's not a new mutation that's occurring. And so basically we think the epizootic just shifted the advantageous mutations that were already there, shifted them higher in frequency. And we can look, for example, at this region around BBX and find that when you compare different time points, especially when you compare the 1990 versus the 2014, you can see a detectable increase in differentiation when we compare 1990 to 2014. And that also coincides with an increase in linkage disequilibrium over these different time points. And these are the kinds of subtle signatures that we're looking for. And I mean, ultimately, I think we're going to need some sort of machine learning method to pick out these regions. But for now, we have a few regions like this that we want to look at in more detail. And so just to summarize this part of the talk, we've done a lot of sequencing of both a pathogen and the host, and we're now looking for very subtle signatures of natural selection in the host birds that have been sampled over different time periods. And so this is ongoing work that we're excited to be continuing with Santiago. Let's now switch to talking not so much about microevolution, but instead talk about macroevolution, distantly related birds. And this is always a challenging scale on which to work because species are long diverged. But we noticed there's a lot of work has been done to use phylogenies to study different aspects of evolution, how traits or characteristics evolve, a lot of use in biogeography. But we noticed, a few of us at the evolution meetings a few years ago in the United States, that there was not much work being done to use phylogenies to link genotype and phenotype. So we call this the phylo-G2P approach, linking genotype to phenotype through phylogenies. And I'm not going to talk about these two examples here, but we've actually been using this approach to look at different kinds of adaptations in birds, including how they taste sugar and where their feathers came from. This is work I could happy to answer questions about. But what I'll tell you about is how we've looked at loss of flight in birds. And this, our approach is outlined in this opinion piece, which came out last year. And yeah, we're very excited to be promoting this new use of phylogenetic trees. Just to give you a little more detail, I mean, most species that you can breed in a lab, you can create a hybrid population. You can make variation is segregating in the population, and you can map the locus using different kinds of techniques. But of course, most of biodiversity we can't breed in the lab, and so we need other approaches. And in this approach, you could imagine finding clades that have a lot of evolutionary transitions of the phenotype of interest, and then using those multiple origins of the trait to correlate with genomic change, basically. And that's the spirit of the phylo-G2P approach. And there's a lot of really cool examples out there. One example which I like is done by Gil Bejarano at Stanford University. He basically took a whole bunch of mammalian phenotypes and a lot of these non-coding regulatory regions in mammal genomes, and he laid them on a phylogeny. And so what he's looking for is a covariation of the phenotypic change and the presence or absence of these regulatory elements. And he basically, in this case, he was looking at the digit morphology on the feet of mammals. And he used this sort of approach that sort of a big data kind of approach. When he plugged it through his algorithm, he came out with several non-coding elements that were nearby to genes that actually have quite a lot to do with digit and phalanx development, okay? The phalanx, of course, is one part of your digit in your hand or in your foot. And remarkably, he was able to pull out these genes simply by looking for associated regulatory elements that were evolving in tandem with the phenotypic trait. This is, I think, a very cool example, and it shows convincingly that this approach can link genes with logical functions to phenotype of interest. Now, in our case, we're going to look at the loss of flight in birds. And these are the group that we're going to look at, we call the Paleognathi, but it's basically, famous group including ostriches and emus and cassowaries from Australia, and rheas from South America, and kiwis from New Zealand. And in the middle, you've got this very interesting group of birds, which we call tinamous. And of course, there are many species of tinamous in Mexico. And they're the only member of this group that can actually fly. And so they're going to play an important role. And as you can imagine, there's a lot of anatomical changes when a bird goes flightless, one of which is the keel, which is usually quite large and which extends on the sternum like that, is no longer present in flightless birds. So that's so that's a lost trait. Often the body size gets very big. I mean, these ostriches and moas that are extinct, some of the largest birds ever to walk the earth. And then finally, you get a lot of loss of elements of the forelimb. So you get loss of digits, and you get loss of entire bones in the forelimb. In fact, the moas from New Zealand have lost their forelimb entirely. They lost all of their forelimb elements entirely. No one has ever found a humerus or a ulna or a radius from a moa. So there's a lot of interesting changes, and we can use the genetic data, but also epigenetic tools like ATAC-seq to figure out where regulatory elements are actually acting. So it turns out that people used to think that these tinamous were a sister to all of the flightless species, and so you can imagine if that was true, that flight might have been lost once at the base of the tree, and that these birds may have spread around the globe, maybe through continental drift. But it turns out that the tinamous, which are the only group that can fly, turns out they're actually embedded within the radiation of flightless birds. It was a very surprising result that overturned decades of study. And it really brought home the possibility that flight might have been lost multiple times, okay? Maybe as many as 6 different times, depending on how you want to interpret the biogeography. And we know that, there's a possibility that if the rate of loss of flight is very high, and if the rate of gain of flight is very, very low, it's plausible that flight could have been lost multiple times. That's what this red region of parameter of character change space suggests, that it's plausible that flight could have been lost multiple times, and that this would actually be sort of a more parsimonious explanation for the pattern than having, for example, a gain of flight in the tinamous again, okay? Generally, people think that is unlikely to have evolved flight a second time within the birds. So we did a bunch of genome sequencing, and a bird genome, as you probably know, is about 1 gigabase. So quite a bit bigger than the Mycoplasma genome, but not so big as mammals. And these days with the next generation sequencing, we can sequence a bird genome for not too expensive. We also inherited a genome from a moa from New Zealand. This was work done by a collaborator who sadly passed away, but it allowed us to study another transition to flightlessness. Now we set up a sort of a database which is publicly accessible and allows you to line up the genomes in different combinations. And it's a nice genome because it's what we call a nice alignment. We call it a reference-free alignment, because instead of using a reference genome to map everything, we can instead use a phylogenetic tree and retain all of those parts of each genome that might not actually map to a single reference. And so this was an important advance that we were able to take advantage of. So the first business was to make a tree, see what the phylogenetic relationships were. And we've used a lot of markers across the genome. These are different. We found, many people have found that non-coding markers tend to be more reliable for making trees than actual coding portions of genes, maybe because genes might often be under natural selection more. And we had some ideas about what the tree would look like. And we were particularly interested in where the rheas from South America were landed, because you can see they're very unclear what they're related to in these different studies. And so here's our tree. It's a very it's actually a very robust tree. Some of these branches look very, very short, but actually they're very, very well supported. Just through a lot of data at this problem, the tree shows, for example, that the Tinamous are embedded within the flightless birds, just like the previous tree showed. And it's consistent with anywhere from 3 to 5 losses of flight, okay? And it's also corroborated by transposable elements. We can use transposable elements to test the branches on this tree, and we are able to recover exactly the same tree. And so we're very excited about this tree, and we think it's probably the most accurate tree of this group so far. Now, just a quick moment to acknowledge one of my graduate advisors, Alan Wilson. Alan Wilson was a famous molecular evolutionist who sadly passed away just as I was finishing up my dissertation. He did quite a bit of work on bacteria as well, although he's mostly famous for his studies on human evolution. And he and Mary Claire King noticed that chimpanzees and humans were very, very similar at their protein sequences. And so because of that, he proposed that maybe it was gene regulation that was very important to the evolution of phenotypic traits. And that's a paradigm which I think is still very, very compelling even today. The problem, of course, though, is that the non-coding genome is very, very complicated. In birds, like humans, the non-coding genome is the vast majority of the genome. It's probably 98% of the genome, and yet we don't know very much about it. And so what we did was to use these regions called conserved non-exonic elements. They're basically like the non-coding elements that I told you about earlier, and use those as kind of a foothold into the regulatory functions of the genome. So these are regions that are highly conserved across species. As you see in this diagram at the bottom, the conservation says they're doing something very important, but they're not a gene, so it's probably some kind of enhancer or regulatory element. So we found a bunch of these around the genome, and we looked at how they were evolving on the tree. And in particular, we developed a method to count essentially the number of changes that are occurring in a given element. Like you see here, we can count the number of changes that we see in the flying birds. Those are the birds down here. You can see there's not many changes occurring there in this element, which is 250 base pairs. Whereas in the flightless species, we see, I think, a detectable increase in the number of substitutions going on. And so we worked, and this could be an indication of a change in function of this non-coding region, or it could be a loss of function. We're never entirely sure, but we developed a very very exciting approach, which I don't want to go into the details now. It's been published last year, or actually 2019, and it's a method for basically identifying these convergent rate increases. So, for example, say we want to find elements that are increasing in their rate in these 4 target species in red, 1, 2, 3, and 4. Most methods that in the literature will tend to identify elements that are also accelerating in these non-target species. For example, these might be our Tinamous. We wanted a method that could distinguish between acceleration in this entire clade versus acceleration just in these 4 target species. And so we teamed up with some statisticians here at Harvard, and we developed a really powerful method that seems to work better than a lot of the methods out there. Anyway, here's an example of an element that is conserved across all of bird evolution. I mean, this is like 100 million years of bird evolution right there. But it's accelerated specifically in these rheas and in the moa species. So that's really interesting. And the fact that it's accelerated in these 2 flightless birds means it might have something to do with loss of flight. So we basically tabulated all of these and found that about 1% of our regulatory elements seem to be accelerated in 1 or more flightless bird species. So this was very exciting. And we can then look at genes that are close to these, we call them ratite accelerated elements, ratites being the flightless paleognates. And you can see here that when we look at genes that have a large number of these ratite accelerated regions, we actually find a number of genes that are very similar to, that are very suggestive of a role in skeletal changes. So for example, HOXD4 is very well studied. TBX5 is very well studied. These are all at DOC1 is known to be a limb morphology regulator. So these are genes that make logical sense. And I should say that we looked at the genes directly. We asked which genes are actually undergoing acceleration. And we did find some genes, but they didn't fall into any particular Go category. They were sort of scattered all over the place. So we really think that regulatory change is more important in this case than change in the actual genes. And just to finish up, I'll tell you a little bit about some of our epigenetic work. I mean, so you can imagine if we find a non-coding element, that's great, but we don't really know where and when during development it would be active. And one way for us to do that is to use a technique like a TAC-seq, which can tell us which part of the chromatin is open or closed in evolution. And we did this. We looked specifically at the forelimb and hindlimb, working mostly with chickens. But we also looked at other parts of the developing embryo. And basically, this method identifies regions of the genome that are unwound and open in terms of accessibility of transcription factors. And we found several regions around the genome that are really suggestive of a role for these non-coding elements. We also looked at gene expression in the fore and hind limb of these various species. And I'll just end by showing you a little bit of data that's not published now, trying to provide evidence that there's been convergent changes in the transcriptome of these birds, as well as in the epigenetic changes. So for example, here is a plot of the number of genes that are differentially expressed between the forelimb and the hind limb of each of these 5 species of birds. And what's interesting is that whereas the chicken and the tinamou both have a relatively small number of genes, those 2 birds can fly. I mean, chickens, they don't fly very well, but they can fly better than an ostrich can. You can see that the number of differentially expressed genes there is relatively small, whereas in the flightless birds, it's quite a bit greater. And so that's exciting, again, suggesting that gene regulation might be important here. And we can also look at the pattern of transcriptome evolution as a whole. And so we can think of the transcriptome as this sort of high dimensional trait. And we can, using techniques from phylogenetics, we can ask whether we see evidence for convergent evolution in the flightless species. And so when we plot out the, here we've plotted out the transcriptomes of several species, but also several stages within each species, several embryonic stages. And what's interesting is that notice how the tinamou points here and the chicken points here, whereas all the flightless birds are clustered in this part of the space. And this suggests to us that there's been this convergent evolution from, these are sort of proxies for ancestral species, but they're both leading to this cluster of points in the flightless bird space. And we think is evidence of convergent change in the transcriptome as a whole. And we actually see very similar patterns when we look at the pattern of ataxic change as well. So I'm actually I'll skip over these last couple of slides and just sum up. And what we've done here is to take a group of birds that are very distantly related, and of course we can't breed them in the lab. And we tried to use new techniques to make connections between the genotype and the phenotype, specifically between these non-coding elements and loss of flight. And we then used ataxic. I didn't show you all the data, but we can use ataxic to basically ask whether each of these elements is active in a particular organ, like a forelimb or hind limb, at a particular time during development. And when we do that, we actually see quite a bit of evidence that these accelerated non-coding elements are undergoing, they're in an active state when it comes to their chromatin state. And so we think they're very good candidates. We haven't figured out the gene that causes loss of flight. We suspect it's probably many genes, of course, very complex trait. But using this phylogenetic approach, we think we've narrowed down the window of genes and regulatory regions that could be very important for loss of flight. And with that, I will end and thank my collaborators here. We're continuing some of this work with some new funding, and we're very excited. Cliff Tabin is someone who he's a developmental biologist, and he's been helping us quite a lot with the chicken studies. And then there's several. Tim Sackton did a lot of the computational work, as did Alison Cloutier. And Phil Grayson did quite a bit of the genome sequencing and ataxic. And in truth, I'm really just the resident bird watcher. I'm surrounded by all these talented students and postdocs, and but hopefully I enjoy the big picture of the project. So thank you very much, and I would be happy to take any questions from you.
Thanks, Scott. The talk was superb. Thanks very much. So, any questions? Let's see. We don't have anything on the chat yet, but well, one. So you can read the questions from the chat, Scott.
Yes. Oh, I see. Yeah, okay, I see. Here's a nice question from Sergio. Oh, yes. So there was, I wonder, Sergio, if Sergio's thinking about a study that came out in maybe 2017. It was on these cormorants. We call them cormorants. And it found, it basically proposed a few genes that were associated with loss of flight in cormorants, specifically in the Galapagos cormorant. You might know that the Galapagos cormorant is a flightless bird. That's an interesting study. You know, basically they didn't even look at the non-coding portion of the genome. So if they found anything, it was going to be a gene. And in our case, we sort of looked both at the genes and the non-coding elements. And although I didn't talk about it in detail, we found a much stronger signal, much more functionally integrated signal with the non-coding regions than with the actual genes. And so we think that the gene regulation is probably more important than the actual genes. We didn't find we didn't find a lot of evidence for pseudogenes. We didn't find a lot of evidence for function altering amino acid changes. Really at the level of the genes, there wasn't a lot of really interesting stuff going on. So again, we think the regulatory change is probably what's driving this.
So related with that question, Scott, obviously you are talking about changes in non-coding regions, but are you just talking about substitutions? And so maybe a more general question is, so this seems to work very well with, let's say, mutation substitutions in genes or non-coding regions, but what about, I don't know, gene content variation, rearrangement, stuff like that?
You mean like rearrangements and stuff, inversions and things like that?
Right.
Yeah, there's been some very interesting work suggesting that chromosomal inversions could be important for some behaviors in birds, and very interesting studies. And yeah, I think you're right. There could be some very interesting rearrangements going on. We haven't looked in that detail. You know, our genomes are pretty good quality, but they're not like chromosome level. And so looking at inversions may be a bit challenging, but it's certainly something that we could try to do. Absolutely. Yeah.
All right. So we have another one from Esperanza. And
okay. Yeah. Hi, Esperanza. Thank you so much for telling me about your very interesting mycoplasma studies. Santiago, we have to compare our mycoplasma to mycoplasma. Are there individual susceptibilities in house finches? Yes, there are, especially from different populations. And so the populations in the eastern US, which have been exposed to the mycoplasma since the mid 1990s, they seem less susceptible. They can still get the disease, but they don't get as sick and they don't die as frequently as do birds from the western US, which have only encountered the pathogen in the past few years. So there's quite a bit of variation, and we'd like to quantify that better and see how it correlates with gene expression. There's still a lot of variation out there, and we actually did some work showing that there seems to be some evidence that the plumage brightness suggests the level of susceptibility. So very red birds seem to be less susceptible and undergo less gene expression change after infection than do yellow birds and more dull birds. And so, a lot of times people have thought that plumage might be an indicator of health and susceptibility. And so we'd like to extend that work, but the initial signals suggest that that might be the case. Here is a question from Ivan. Let's see. Genetic diversity between finch populations. Do you think the differences in genetic diversity is big? What do you think about the Lewinton's paradox? Okay. Oh, this is great. We should have a beer and talk about Lewinton's paradox. I think I think Lewinton's paradox is let's see if I get this right. It's the idea that the amount of genetic diversity in a species may not be able to maximize because of sort of recurrent selection going on. In other words, when you look at genome diversity across many different species, it tends to fall in a very narrow window. It doesn't seem to correlate with the actual population size, right? We expect big populations to have more diversity than small populations. And that's Lewinton's paradox. And yeah, that's a great question. And you might be familiar, Yvonne, with there's a nice paper by Dan Hartle's group back in 2015, I think, in PLOS Biology, where they developed this really interesting model showing that, trying to quantify the effects of how natural selection could be depressing genetic variation in different species, especially species with very big populations. I think that in the case of the house finches, it's relative, right? In other words, even if genetic diversity in the house finches is being depressed by selection, it's not reflecting the actual population size, still we see a decrease in the introduction, and we also see a decrease after the epizootic. So even if those levels are not reflecting the true genetic diversity, we think relative to each other, they probably are giving us a good signal. But I'd be curious to hear what you think about that. Let's see, Jocelyn has asked, did you find some genes or regulatory regions involved with the hyperglycemic condition in birds? Oh, very interesting. Now, hyperglycemia, I'm trying to think, this is the fact that they have a lot of sugars in their blood. I'm trying to make sure I have the right phenotype here. We have not done a lot with bird physiology and nutrition. I think that would be really, really interesting. I did read a recent paper which seemed to suggest that the way birds regulate the lipids in their blood could have a very interesting genetic basis. I haven't seen much or done much looking at hypoglycemia, but I think it's really an interesting possibility.
Okay, so I have another two questions, Scott.
Yeah.
So the first one is thinking about those Arizona and Alabama populations. So are you talking about the same genotypes, or did you find different lineages there?
You mean in the Mycoplasma?
Yeah, exactly.
So Santiago, it's sad, we actually don't have any strains from Arizona, from the Western U.S. I think maybe they have some strains at Cornell or with this Stephen Geary in Connecticut, but that's something that we should do, because I mean, my guess is that Mycoplasma is in Mexico, and I think we could get a much better understanding if we looked at Mexican birds and tried to isolate some mycoplasma from them. So there is some suggestion that the strains that made it to the Western US, it was just a single strain. So just one strain that spread across the whole West. And I think the Cornell group may have published that. But it needs to be confirmed with greater sampling. There's just been way too much actual sequencing and not enough actual field work to collect these samples. So.
And I mean, the other one, I mean, and this is related to the soft sweeps. So have you looked at the other hosts like turkey or chicken to see to those regions and see if also there you have signals for the soft sweep?
You mean, wait, you're saying in the mycoplasma or?
Yeah, in the mycoplasma. No, I'm talking about, I'm talking about the birds, yeah. I mean.
The flightless bird?
Yeah, exactly.
Have we looked at chickens and turkeys for some of those non-coding regions?
No, I'm talking about the stuffed sweets, but I mean, you look just as the house finches, right? But what about if you were to look at the poultry? Because I mean, there is you know what I mean?
Yeah.
So you have a bigger population.
Well, yeah. I mean, I think no, absolutely. I think the CRISPR system is going to be much more variable. I think we probably, as that one graph showed, the diversity in the house finch is so much smaller than in the poultry. So it would be more interesting, I think, to look at chickens, and that should be they should be fairly easy to collect as well. Yeah, I think that would be really fun to do, but we just haven't done it.
Right. There's another one there.
I'm just reading some more comments. I see another interesting comment from Jocelyn about flightless birds seem to have normal glycemia levels, whereas the rest of the birds are considered. I did not know that. I'll have to. Maybe we can follow up after by email afterwards. That would be a lot of fun. And then Yvonne is asking again, right, life history and ecology could have a big effect on reducing genetic diversity. Yes, I absolutely agree. And is it possible in the finches? Oh, is that possible in the finches in resistance to the pathogen? Well, I mean, we know that the fact that these birds, these house finches, they come to feeders a lot. We know that the mycoplasma is spread just by contact. It spreads very quickly. And they're often a flocking species. So there's a lot, it's spread incredibly quickly. There's not a lot, they're not really unusual, I would say, in terms of their life history. Like most birds, their generation time is about 1 year. They have a clutch size of, probably 5 or 6 eggs. And so there's nothing really unusual about their life history. And so, but it is a big puzzle as to why the mycoplasma is only found really in the house finches outside of turkeys and chickens. Very occasionally it's found in these yellow birds called goldfinches. Actually, one of the strains that we're sequencing, I think Santiago, is from a goldfinch. But other than that, they're not found in any other species of bird. And so, at least at a high enough level that we can detect. And so that's a real puzzle. And maybe it's some difference in the immune system. I really don't know. It's a very interesting question.
All right, I guess the time is up. Thanks very much, Scott. We really appreciate this, and thanks again.
Thank you very much, Santiago. And I look forward to visiting you guys, maybe when the pandemic is over, reconnect. Thank you very much for coming to my talk.
Thanks very much. Later.
Bye-bye.